Bismillahirrahmanirrahim. My name is Taram Fatma and the topic under discussion is gene expression in prokaryotes and eukaryotes. To have a complete understanding of this topic, we have to understand some important terms which are prokaryotes, eukaryotes, and gene expression. So, what are prokaryotes? Prokaryotes are those organisms which do not have well-defined nucleus and membrane-bounded organelles in their cell, whereas eukaryotes are those organisms which have well-defined nucleus and membrane-bound organelles in it. Gene expression. What is gene expression? Gene expression is the process by which the information stored in DNA is transferred to a functional product known as protein. Now, gene expression in prokaryotes. It comprises upon transcription and translation. Transcription. Transcription is the process of conversion of double-stranded DNA into a single-stranded RNA. Single-stranded RNA, and translation is the transfer of RNA into the proteins. Now first we discuss about transcription. The steps involved in the transcription of prokaryotes are initiation, elongation, and termination. And the machinery utilized in transcription is RNA polymerase. RNA polymerase have 5 subunits: beta prime, beta, alpha 1, alpha 2, and mega. When the RNA polymerase have these 5 subunits, it is known as core enzyme. And when the sigma factor gets attached with this core enzyme, it is known as holoenzyme. As the RNA polymerase can't start initiation on its own, it requires the help of a sigma factor. It is a protein which allows specific binding of RNA polymerase with the promoter, so the complete RNA polymerase has 6 subunits. In the first step, which is initiation, RNA polymerase binds to the promoter region. The DNA at this stage is still in the double-stranded form, and in the next step, the strands near the transcription promoter site unwinds. As the sigma factor releases, the RNA polymerase proceeds for the process of elongation. As the genes are transcribed, the transcription process must be stopped, and which is known as termination. There are 2 methods of termination in the transcription of prokaryotes, which is row-dependent termination and row-independent termination. Row-independent termination. It does not require external proteins or factors for termination to occur. It is also known as intrinsic terminators. When RNA polymerase transcribes the sequence, the resulting RNA forms a stem hairpin loop-like structure by base pairing it with itself, and the hairpin loop halts the RNA polymerase enzyme, and the loop is followed by AT-rich sequence. And after transcription, which becomes AU, as they are the weakest, as a result transcription occur. Now row-dependent termination. It depends upon a protein known as row factor. Row factor is an ATP-dependent hexamer alkali, and the row protein binds in a cytosine-rich site known as Rut site. When RNA polymerase reaches 100 nucleotides away from the right side, it stops the transcription, and the sequence that holds RNA polymerase is known as row-sensitive site. Row protein utilizes the ATP and translocates at the RNA-DNA hybrid and unwinds the region. Now move towards the translation. Translation is the conversion of messenger RNA into the proteins. The smallest subunit gets separated from the largest subunit with the help of 2 initiation factors known as IF1 and IF3. This complex then binds the purine-rich sequence known as Shine-Delgado sequence. The Shine-Delgado sequence is paired on the complementary sequence on 16S ribosomal RNA on smaller subunit. It shows that the smaller subunit is on the right place. Another amino acid, IF2, brings in initiator transfer RNA with the amino acid, and the larger subunit will join the complex and initiation factor are released. The ribosome has 3 sites: A site, B sites, and E sites. A site is the entry site for the new transfer RNA. B site is occupied with peptidyl transfer RNA. It carries growing polypeptide chain and E site is the exit site for the empty transfer RNA after it's done with the delivering amino acid. The new transfer RNA carrying amino acid enters the A site of the ribosome. The anticodon of transfer RNA is complementary to the codon of messenger RNA. A peptide bond is formed between two adjacent amino acids. As peptide bond form, amino acid at P site released and the empty transfer RNA moves towards E site. A site becomes empty as ribosomal subunit moves one triplet towards messenger RNA and the empty transfer RNA released from the exit site. And the cycle is repeated for each codon. The termination happens when one of the three stop codon is positioned in A site. At that point, no transfer RNA can fit at this A site. Instead of these codons recognized by proteins, they are recognized by the release factors. Binding of release factor catalyzes the cleavage of bond between polypeptide and transfer RNA, and the polypeptide chain is released and the ribosomal subunits are dissociated into the larger and smaller subunits and ready for the new mode of translation. The transcription in eukaryotes. DNA strand has both promoter and terminator regions. Promoter region of a gene acts as recognition site for RNA polymerase to bind. This is where the majority of gene expression are controlled. Binding of RNA polymerase causes DNA to get unwind and become open. During elongation, RNA polymerase slides along the DNA strand. As complementary base pair up, the RNA links nucleotide to the 3' end of growing RNA molecule. Once the RNA polymerase reaches the terminator region, messenger RNA transcript is released. Messenger RNA transcript includes exons, which is the portion or the region of the messenger RNA which codes for protein, which is a coding region, and non-coding region called introns. The post-transcription modifications occur as the 5' cap and poly A tails are added and the introns are removed. It is called intron splicing. Now the nitrogenous bases are grouped into the 3 letters called codons. Translation begins when the messenger RNA start binding to small ribosomal subunit of upstream of start codon. Each amino acid is brought to the ribosome by specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of transfer RNA. The complementary base pairing occurs between the codon of messenger RNA and transfer RNA molecules bind to the start codon. The larger subunit binds to form a complex. The ribosomal subunit has 3 sites: A site, P site, and E site. During elongation, individual amino acids are brought to messenger RNA strand by transfer RNA molecule. A charged transfer RNA molecules binds to the A site and peptide bond forms between its amino acid and are attached to the transfer RNA molecules at P site. Complex slide down one codon to the right where the new uncharged transfer RNA molecule exits from the E site and the site is open to accept next transfer RNA. And the elongation will continue until a stop codon is reached. The release factor binds to A site at stop codon and the polypeptide is released from transfer RNA and the complex dissociates. Jazakallah.